Hi everybody, we're going to get started so you can get seated. I'd like to welcome you all to the inaugural New York Sea Grant hosted Oceans and Atmosphere Colloquium. We came up with the idea of New York Sea Grant hosting one of the Friday seminars each semester to feature a current or past New York Sea Grant funded researcher or current or past Sea Grant scholar. For those of you who are not familiar with New York Sea Grant, it's part of the National Oceanic and Atmospheric Administration, and it is a grant funding agency that also does extension, marine science extension. There's 30 Sea Grant programs throughout the country on all the coastal states and Great Lakes states. And it got started in 1970 with the passage of the New York Sea Grant College program. Anybody who wants to learn more, you can come see me afterwards. So for our first New York Sea Grant hosted speaker, we have with us today Dr. Daniel Molloy. He's the aquatic invasive species biologist who has been with the New York State Museum for many years, recently retired. He was located at the Cambridge Biological Field Station in Cambridge, New York, in lovely central New York State, just north of Albany. And he's now at the University at Albany as an adjunct professor. He got his PhD from SUNY Environmental Science Forestry, and from there he went to the New York State Museum and Biological Field Station at Cambridge, ESF. So today, New York Sea Grant funded Dan on zebra mussel biocontrol back in a project that ran from 1999 to 2002. And the project was looking at using bacteria for biological control of zebra mussels. And Dan has done a lot of research on biocontrol of dreissenid mussels and other invasive species. For those of you who don't know, dreissenids mussels, the zebra mussel, and Quagga mussels are exotic species that have invaded Great Lakes and Hudson River and have been a costly invasion. And today, Dan is going to talk about research regarding developing the next generation of environmentally safe biocontrol of zebra mussels. So, did I leave anything out?
No.
All right, Dan, take it away.
Lane, thank you very much.
You're welcome.
So we have an alumnus there from ESF. Good. The title has 2.0 up there. That's my corny attempt, to say it's a new generation of something and pique your interest. And it also says, because the title has 2.0 up there, that's my corny attempt, to say it's a new generation of something and pique your interest. And it also says, because Dan is also known as Corny Dan, outside the shell, not outside the box in this case, because we're talking about dreissenids. So I want to get started. There may be people in the room who are not familiar with dreissenids. Lane did talk about them. So I just want to introduce the two species that are in North America. There's about a half dozen that are still existing, and two, in Eurasia, and two of them have come to North America. The zebra mussel and the Quagga mussel are their common names, so they're in the same genus Dreissena. So I've introduced the two characters I'm going to talk about, and again, for those people, and my talk is also being videoed, so there's an audience in the future, and I want to spend a little time for maybe those people who don't know about zebra and quagga mussels as an introduction. And maybe some of you here, because you're marine and not freshwater, have not seen photos of them. But they basically, both came in the 80s into the Great Lakes and have spread from there. And because they have special abyssus like the blue mussel, they are the only freshwater bivalves in North America with abyssus of that size. And they can attach like mittless to almost anything. So this is my slide to show that there can be impacts in terms of industry. You see that pipe in the lower right corner. So industry has had to deal with them. And boats, you see, you go over to the left there here, looking at that propeller, boats are spreading them from lake to lake, and they have an ecological impact on lakes. And you can imagine shells wind up on beaches, and people step on them when they're attached to rocks. So they have, recreational, ecological, and industrial impacts. We would have been so much better off if they had never come. And here's that byssus that they have, like your blue mussel, and they are the only species now in North America with that byssus, and allows them to attach inside pipes onto rocks, etc. Now, I've introduced the two invasive species I'm going to talk about, but I want to add just a little bit to what Lane said about who I am. And as it says there, I'm a scientist who's attracted to looking for solutions to pest problems that have been considered by many to be unsolvable. Hello, my name is Dan, I'm a masochist, okay? So I tend to take these things on, and I'm sure you've done the same in many ways, and you're at night, you're staring at the ceiling saying, why have I done this? Where's the progress, but that runs through my core, to pick projects that are very challenging, and hopefully not too much of a dreamer. I've got some successes to show you to gain some confidence in my credibility, not to build up my ego, but I want to show you some successes along in my career where I've taken on problems that other people said, that's a needle in a haystack, he will never solve it. You look at the bottom of that slide, you can see the thread running through my career. It's not just any old pest, aquatic pests, and actually freshwater pests. And at the very bottom, I've used my expertise in invertebrate pathology to find these solutions to these aquatic pest problems. So what have I built upon? What I want to show you, what have I accomplished, my lab has accomplished? I want to show you examples of what I would call biocontrol 1.0. I'm giving you a couple of examples of those successes, and then you can see how, when I finish my talk in the second half, and I talk about biocontrol 2.0, you can contrast those two biocontrol approaches. So, the first project that I worked on as a grad student at the College of Environmental Science and Forestry was how to control black flies in the Adirondacks. And people said, you're just not going to be able to do it. These flies are going to continually be around our heads. If you're not familiar with black flies, especially in the Adirondacks, you know that they're biting and they're hovering, can cause problems. There's a female there on human skin. Well, a scientist in Israel found a bacterium, a subspecies of Bacillus thuringiensis. If you're an organic gardener, you may know that bacterial species, Bacillus thuringiensis, BT, very, very safe to use. This scientist found this strain in Israel in the desert, and it was shown to be toxic to mosquitoes. You could kill mosquito larvae by feeding them the bacteria. And then we did tests along with other collaborative labs that showed you could kill black fly larvae. And the way the mortality occurs, again, so we're talking about a subspecies of Bacillus. Bacillus thuringiensis is the species. Israelensis, of course, found in Israel. And so this is the first project I'm going to show you that went all the way to commercialization, a biocontrol project. And the way the mode of action here is, as that vegetative cell is growing and producing the spore, if we look down here at number 5, the spore, there are also parasporal crystals that are produced. When that vegetative wall of the bacterium disintegrates, releasing the spore, it also releases these parasporal crystals. And so there's the parasporal crystals. And the black fly larvae in streams and rivers, that's where they breed, catch these crystals on their cephalic fan. So here's an illustration. Here's the real thing. The head of a black fly larva, catch them and kill them. So it was an amazing success in environmental control of black flies in the Adirondacks. And it's still going on after several decades. Thousands of miles of stream are being treated with this method. I show you some people, they measure the flow of the stream, they apply the bacteria, the black flies are killed. Extraordinary selectivity. That's Biocontrol 1.0. It's not infecting the black fly. It is actually dissolving their midgut epithelial cells, those parasporal crystals, okay? Now, so that's a success in Biocontrol 1.0 that I participated in. I didn't lead that project, but in North America I played a significant role. So I was very, very familiar with how you can take natural products from microbes and use them for pest control and be very effective and extremely environmentally safe. Unbelievably narrow window of organisms it would affect. So I knew that. Then, in 1989, when zebra mussels first started coming in to New York, the Department of Environmental Conservation contacted me, New York State Department, and said, Dan, do you think you could find a different bacterium, not BTI, that zebra mussels could be controlled with? Because the power companies, so they said there's zebra mussels coming, the power companies need something besides chlorine, bleach, essentially what they're putting in their pipes to control. And they have grant funds for you to look for it. And I said, no, wait a minute. My PhD is in entomology. I've never worked on a mollusk before. You know, when you get out of your comfort zone, right? But I said, hey, and the power company said $800,000 to work on this project. They came, of course, in a series of fundings with good progress reports. So, my contact at DEC, he said to me, Dan, look, this is an aquatic organism. You work with aquatic organisms. Yes, okay. But it's not a bug. It's not a bug. No, don't worry. It's still a filter feeder. So you can see here in this photo, there's the siphon, the inhalant siphon of this mussel. So Dan, it's the same concept. You find a bacterium which serendipitously, not by evolution, but serendipitously has natural products in its cell which can be used for pest control. I said, well, you have the funding, I will try. 4 years of taking their money. I had successfully identified 700 bacterial strains that didn't kill anything. And I went to the chair of the funding committee in this New York State power pool that was funding me. And I said, I don't think I should continue to take your money. And again, we had time on this contract. And he said, why? I said, I haven't found, I believe it exists, that we can find a soil microorganism. Its niche is something else, but it has natural products which can be used. He said, go back to your lab. You know, you hear these stories and you go, that's baloney. Well, you're hearing it right from the horse's mouth. Go back to your lab. If they're spending their money for 4 years and getting nothing. Go back to your lab and keep looking. 2 months later, we found a strain of one of the most common bacteria that exists. It's Pseudomonas fluorescens. Its niche in life is to live in association with plant roots, and it produces secondary metabolites which inhibit mildew, fungal infection of those roots. But it just so happened that this isolation was a strain of Pseudomonas fluorescens which could kill zebra and quagga mussels. So we immediately said, hey, let's patent the species, Pseudomonas fluorescens, patent the species for control of zebra and quagga mussels. And the patent attorney said, well, how many strains have you tried? We've only tried one. We'll try some more. We tried 9 other strains. What do you think happened? 0. None of them, so there's this particular strain is the one that went on to be the commercial product, and you could only patent the strain because it was unusual. You couldn't patent the species. So I learned a little bit about patent law, and there's the 1x2, 3-micron comma-shaped Pseudomonas fluorescens. So, and by the way, it's patented. I will always be the patent inventor. You folks at Stony Brook University have a patent policy, right? Do I say that? You have a patent policy? I worked for the New York State Museum in Albany, no patent policy. So they shook my hand and they said, you are the first patent we've ever had, and we are the new owners. Now, I had a full-time job, and I wanted to keep it. So I received no royalties. The ownership has been transferred to New York State, the State Education Department, but I will always be the inventor. And I take some pride in that the project worked out. Okay? And I wanted to show you this slide because probably going through your mind saying, okay, you know, you can kill mollusks with a lot of things, but then you're trying to be environmentally aware and conscious. Shockingly, the first strain that killed zebra and quagga mussels, that strain that I just showed you, also had incredible selectivity. So there are things it can kill, but I can blanket tell you now, if you are going to use a commercial pesticide for controlling zebra or quagga mussels, the most selective one is this bacterial strain. Okay? So we did achieve it. I'm trying to show you. We did a lot of testing against fish. We did a lot of testing against other bivalves. No mortality. And USGS now in La Crosse, Wisconsin, is doing a lot more testing. Not just testing against adult freshwater clams, unionids, but also testing against juvenile stage, Glaucidia, etc., to define the environmental impact. There will be. Okay, there'll be some environmental impact. So as its patent inventor, I take great pride, as it says there, that my team came up with something that's eco-friendly. It's very selective. And it's called Zequinox. That's what the commercial company gave it as a trade name, product name. So, but the talk is not over, because this is all biological control, what Dan calls 1.0. It's like there are commercial products, but they're not good enough to control zebra and quagga mussels in entire water bodies. This stuff is great. You got a power plant. That's why my team worked on it, and I invented it. That's what it was designed for. Treat power plants. Don't use these broad spectrum chemicals that they put in their pipes to clean them out. Use a product such as Zequinox. You've got a choice now. But what happened in the last couple of years, once the Zequinox became commercialized, I started getting calls. We have a lake. We want to control zebra and quagga mussels in the whole lake. And I said, you can't. It's too expensive. It's technically unfeasible to try to get every nook and cranny in a lake imbued with some chemical or biological Zequinox that's going to kill zebra and quagga mussels. So technically, it's almost impossible. I'm talking about the whole lake. Financially, it's prohibitive. I can tell you that the Bureau of Reclamation wants to treat a reservoir in California. It's several hundred acres. That's not a very big. In this reservoir, they draw the water down. It'll be 700 acres. They've estimated the cost to try this at $3 million. But you're not just buying the product. There's all sorts of ancillary costs involved in trying to treat an entire lake. So, here I am. I've given this talk before, not this exact one. The electrons are still moving. As I said to Lane, I said, I've got to keep tweaking. I want to get this talk ready for this audience. So, here I am to introduce to you my idea for Biocontrol 2.0 so that zebra mussels, quagga mussels, we'll call them dreissenids, can actually be controlled throughout an entire lake. Because what we have commercially available is not going to be used. Too expensive or technically unfeasible or environmentally unacceptable. However, you have to put this into perspective. These mussels, they've been here for 30 years. They came in the mid-'80s, let's say. But in Western Europe, they've been there hundreds of years. Their native range tends to be Eurasian area there, okay? So they went through Western Europe starting in the early 1800s. So they've been there for a couple of, no one has ever tried successfully to control these things in an entire water body. So here we go again, Masochist Dan, another needle in a haystack project. So remember I said that you can't control these things in a whole lake. It's not economical. That's what I tried. It's economically prohibitive. That's what I try to say in this sentence here. No organization has the money for lake-wide treatments and retreatments year after year. Even if they do it every 2 years, lake associations do not have the money. In fact, the Bureau of Reclamation hasn't treated this several hundred acre water body in California. They're trying to find the $3 million. The mussels have been there for over 5 years, easily. So it's economically, you're not going to use currently available molluscicides, whether they be Zequinox or not, to control an entire lake. So here's Dan going out on a limb. The control agent ideally for whole lake treatments, it must be applied only once in a small area of the water body. It must be self-perpetuating. You apply it once in a small area, and you don't come back for several years, or maybe not come back at all. It's going to be a very tall order. It is not only self-perpetuating, but where you applied it, it has self-spread. Well, what's the only thing that's going to be able to do that? It's got to be something that's alive. This has to be a live biocontrol agent. Now, if there are people like myself in this audience, you know, the hairs may go up on the back of your neck saying, oh yeah, we're going to apply something live into a lake? Haven't a lot of mistakes been done in the past? And the gentleman here is chuckling, okay? Absolutely. Absolutely. A lot of mistakes made because people didn't do science comprehensively, okay? So, that's what Biocontrol 2.0 is. Now, I'm, again, an environmentalist. Above all, whatever this live organism is, it must be extremely selective for infecting dreissenids. And, again, I'm a biocontrol kind of guy. Am I going to use predators? Am I going to use parasites? Parasites. My focus is on parasites because they can be very host specific. There are parasites, I've been studying them for 20 years of these dreissenids. You can only find them in dreissenids. They appear to be obligate host specific to dreissenids. So I've got some candidates in mind, but predators, in my review of the literature, there are no predators of dreissenids that are specific to dreissenids. So I'm going the route of parasites as the candidate. So here we come to the main focus of my presentation. What is this Biocontrol 2.0? And why is it different? Like I say here, look at this statement. What I'm going to talk about is a game-changing, bold, new paradigm for dreisenid control with an enormous potential. A game-changing, bold, new paradigm? There's probably some people here saying, Dan, you're talking like classical biocontrol. This is not new. You know, the books on that were written, you know, centuries ago, biocontrol. But I've got an outside the shell twist on that approach. And I will take you through that. So I do believe if I'm successful in this project, this is going to be economical. Diana, a colleague of mine, Diana Padilla, is like, she's shaking her head too, I think, here, you know. So, but I think she knows me, and she knows, I hope, that, and that's why I showed you those other successes. I'm not some Don Quixote who has come to Long Island, you know, with some unattainable dream. This is possible. This is possible. Okay, so it's going to be economical. It's going to be elegantly environmentally benign, since this parasite is going to be so host specific to dreissenids, and it really, this is a bold thing. This could take off in North America, causing drastic reductions in dreisenid populations throughout water bodies, to the point, I mean, this is really where zebra mussels and quagga mussels may not be termed invasive species in North America. Invasive species here can be endangered in their homeland. That's not the case with dreissenids, you know. But this has potential to be, and I'm going to give you some examples of this. Now, I'm showing you a tree. What does that have to do with this talk about dreissenids? That tree, the American chestnut tree. When's the last time you saw an American chestnut tree? Gentleman in the first row who was chuckling, okay, you know, smiling, I'll say, okay. Yeah, you don't find them. And why don't you find them? Because in the 1900s, about 100 years ago, I think, fungus came in from Asia, a fungus which is a parasite of Asian chestnut species, came in, wiped them out. They have never recovered. There are lots of selection experiments going on now, trying to find new strains that are resistant to the chestnut blight. Okay, so what I'm trying to show you now, I'm showing you a tree. You know, how about the elm tree and Dutch elm? I'm trying to show you a very specific parasite. Unfortunately, and another, that's a fungus, came in on the backs of bark beetles or whatever, and it's difficult to find an elm tree. So parasites can be very specific and cause drastic reductions. That's my simple idea I'm trying to present to you. You may say, great, Dan. You're giving us examples of trees. This is a talk about mollusks. Now, I'm jumping ahead in my slides here, but in those two examples, I want to give you the terminology used in this area. The trees would be considered naive hosts, and you may know that terminology. Ready? And the parasites that came in, the fungi, are novel parasites. They did not co-evolve with that particular species of fungus, and that was their downfall. The parasite was what is called hypervirulent towards that host. Host did not have the defenses. And that's what I'm saying here. When you have novel parasites, you see the little bullets, they have not co-evolved with the host that they're decimating. The host that they're decimating has not developed an immunity over time to them. And why is that? Because they often have been geographically separated for millions of years. So here's the thought I had a few minutes ago. I jumped ahead in my presentation. If I could show you an example of a bivalve that is naive, that has been documented to be naive, and the population crashed, then maybe you'd even have more confidence that Dan is not just some Don Quixote who came to visit, you know, on a Friday to give the SoMAS presentation. You all know this bivalve, eastern oyster. You know how the industry was booming before. In the 50s, this MSX came in. Now, if you're atmospheric scientists, maybe you don't know this, but in the 50s, the population of the eastern oyster crashed in Chesapeake Bay, Delaware Bay, and if they looked in the oyster tissue, they found these spores. That's why they called it multispore X unknown. And they gave it a species name, Haplosporidium nelsoni. So they gave it a name, but they said, how the heck, what would explain this population crash in the 50s? Well, now we know why it crashed. Tie this into my talk, my mentioning those trees. Why did their population crash? The eastern oyster, Crassostrea virginica, was a naive host to this spore-forming disease of the Pacific oyster. So this disease, which is normally prevalent in this distant oyster species, the Pacific oyster, got into the Atlantic coast, and the eastern oyster was naive to it and crashed. So I'm trying to show you that this stuff has actually happened with bivalves unintentionally. What I'm going to do is, and I'm working on this project, is to do it intentionally. So that's the bold new paradigm for this biocontrol approach. This is the twist. This is the twist. Introduce a novel parasite to our North American dreissenids that they will be naive to. So I am not suggesting following the traditional approach, oh, let's go to the native range of our problem dreisenid. I've done that for 20 years. I found all sorts of parasites, but I didn't find really any that looked like a great candidate, a hypervirulent parasite that could be considered for biologic control in North America, okay? So for 20 years, as I said, as I initially have done on this project, no, that's not shrewd enough in picking a biocontrol agent. Instead, take this outside the shell approach by searching for candidate parasites in the native range of geographically distant, closely related dreisenid species. Don't go to the native range of the zebra and the quagga mussel. Go to the range of some other species of dreissenids. So I've drawn this crude circle this morning, you know, and this is the area that I concentrated on since 1993, looking at parasites of zebra and quagga mussels, and found, you know, quite a few, quite a variety, but none that really were, oh, this is hypervirulent, let me try it. And now I know why. Probably one of the reasons is they co-evolved with these parasites. So they were not hypervirulent. So where is my focus now? It's over here, in the Balkans and Asia Minor and into Asia, looking at species, not of the zebra and the quagga mussels, but related species. So here's the same kind of a map. So I'll bring you down here. There's a Dreissena presbensis. That's not zebra or quagga. It's in Macedonia, Albania, and Greece. That's where I have been sampling. There is Dreissena blanchi in Greece. I've got samples of that. I have not gotten to Turkey yet down here and this species. Okay, so that's the goal. And there's one lake in Macedonia that I've done most of my sampling in. This lake only has one species of Dreissenid, Dreissenid presbensis. There it is, okay? And I've already found a host of parasites in that species. So I'm just going to draw your attention to, I didn't label some of these, it's not worth it, but you get lots of different kinds of protozoans. Here's a spore former that, it's actually, we described it as a new haplosporidium. So I observed these species in Western Europe in Dreissena, let's say polymorpha, the zebra mussel. And then in Lake Ohrid in Macedonia, in Dreissena presbensis, I am seeing very similar organisms. And it doesn't surprise me, but when you sequence them, they're not the same as in Dreissena polymorpha, the zebra mussel. So what I'm trying to say, I'm actually making some progress, because now I've got parasites from Macedonia that are very similar to the parasites that infect zebra mussels in Western Europe and to some extent in North America. But those parasites of the zebra mussels, they're not hypervirulent. I am dying to try a similar species of parasite with zebra mussels. So you see, that's the goal, that's the twist of this biocontrol idea. So when I give talks like this, people say, well, gee, is all your work going to be overseas? And I go, no, we have work to do here in North America. Because if you are going, let's say I'm in Macedonia and I have the parasite and I get the proper permits, etc., and I bring in dreissenids polymorpha to this lab, and I take the parasite from the Macedonian species of dreissenid and I want to try to infect the zebra mussel, I have to know what's the infective stage. I have to know that life cycle so well. So here in North America, there are some obligate parasites of zebra mussels. So work should be done to better you have to understand the life cycle of the parasite before you ever try to have it jump to a different species. You have to know what the infective stage is. So that's work that can be done here in North America at a university, because some parasites that are obligate to dreissenids polymorpha, the zebra mussel, are present. You get a lab colony going, and then you get to know that parasite well enough, you go to Macedonia and you do a lab experiment. So examine host specificity also. For example, here in North America, is that parasite of zebra mussels, which doesn't cause much damage to it, is that going in anything else? We've sequenced it, we have a primer, we can find it in other organisms if it's present. So that's work, again, that can be done in North America. So here's the final thing, because I know I've been talking for 40 minutes. Okay, and I'm getting laryngitis, okay? And I want to end this talk. I hope the concept of Biocontrol 2.0, using a parasite not from the same species, but from a distant species, is in, I've got it, beat it to death, and you understand what I'm talking about. But to end this, I want to give you the 10 top characteristics of the most ideal parasite. And I've kind of taken the lead from that late night show, David Letterman. He had the top 10. I don't know if people stay up late, probably writing papers and stuff, not watching TV. Okay, but so I want to give you my top 10. Again, the 10th is less important than the 9th. It's still important. Okay, number 10. I want to come up with a parasite that can kill both quagga and zebra. I mean, that's kind of obvious. I don't want to have to develop 2 parasites for Macedonia. I'd love to have one knocks down zebra and quagga. Number 9. It is an obligate parasite. It's not a facultative parasite, which under certain conditions can be free living. You release the thing into a lake, it kills some mussels, it's great, and then for several years it's decided, I'm going facultative now. No, I want an obligate parasite, not a facultative one. Number 8, it's easy to mass rear. You got to inoculate lakes, so it's got to be economical enough to mass rear to inoculate lakes. Easy to mass rear. Number 7, it is easy to infect the mussels under a wide range of environmental conditions. That'd be important. Like, oh, this only works in cold lakes. It only works in lakes of 8.2 or above. No, I want something very, very robust in terms of its environmental constraints. Number 6. Now, this may be a little hard to understand, but here's the point. I would like a close cousin of this parasite to be present already infecting North American dreissenids, because it's here, it's here, and you can do so much work on it before you try to do all this expensive work overseas. So if the parasite is here, you can study its life cycle, you can see its host range here in North America, even though it's not killing the zebra mussel maybe. So that's another important. It is lethal and rapidly so. I don't want a parasite, well, you put it in the water and 5 years from now, the mussel will die, That's going to be causing ecological havoc still for 5 years. No, I want a lethal and rapidly so parasite. Number 4, it's self-perpetuating. Obviously, you treat a water body, and you get like 99.99% kill in this small bay that you've inoculated, and you never see it again. So you want something that's self-perpetuating, because people are going to have to pay money to get this parasite to put it in their water body, but I want that to be the last time they ever sign a check. You see, I want it to be self-perpetuating. Number 3, these are all more and more important. It has to be self-spreading. You go to the water body, you put it in, it kills 99% of the mussels within 2 days, and it never spreads anywhere else in the lake. It's got to be self-spreading. Okay, very, very important. Number 2, yes, all of those things are important, but I don't want 2% kill. It's self-spreading, it's self-perpetuating. I want 99% kill, hyper-virulent. See, that's what that'll do. Now, number 1, what I will ask you, esteemed academic people, what is the number 1 characteristic, the most important of this parasite? What is it? Who said that? Did you raise your hand? Yes. Health. This whole parasite is right down the toilet. If you don't have that, that's the whole reason. That's the most important reason, the most important characteristic. We want an environmentally safe way to control dreissenids. There's simply nothing more important as the characteristic. Okay? So the take-home message, my last slide. Okay? Then we'll have a few minutes maybe for chat. Will there ever be lake-wide biocontrol of dreissenids in North American watercourses? Don't give up. Don't give up on the use of parasites for that purpose. Everyone here is scientists of one sort or another. You can never say never. You can never say, I am going to succeed 100%. Okay? But please don't give up on this concept. 10 years from now, 20 years from now, you see something about dreissenids and a parasite, and there's a guy's bald, he has no hair, and a little line says, Dan Molloy. Oh, I remember that talk. I remember that talk in SoMAS. Okay? This is possible. You know what it depends on? Resources and money. Hello, am I telling you guys anything you didn't know already? It depends on funding. Okay, but please don't give up. And the outside, I'm beating you to death with this, but that outside the shell idea is to control a North American dreissenids population. It involves a parasite that they've never encountered before. A parasite from a distant related dreisenid species found outside the native range of zebra mussels or quagga mussels, for example, maybe one from Lake Ohrid in Macedonia. Thank you very much. Thank you.
All right, we'll take questions.
Yes.
How was your approach different when you found the Zequinox bacteria? Did you have the same sort of look at parasites of the species, look at parasites of relatives approach, or was it?
No. With the Zequinox project, we took, that's why I wanted to show you the black fly one. We took the model that was successful in controlling black flies, finding a microbe which had no evolutionary history with the black fly, but serendipitously killed. So the power companies funded me to develop a biocontrol agent which was the same, that it would not be a parasite of zebra mussels. I was doing that project on the side, okay? But it would be just serendipitously a microbe, and after 700 screenings, 700 microbes that never met a zebra mussel in their life, one of them, luckily, by the way, just 2 days ago, a Chinese scientist shared the Nobel Prize for medicine. And I see some folks shaking their head. You know what the topic was? Natural products used for malaria protection from a plant. And I read that article and I said, yes, it can work. And you've heard stories like that. Natural products can be used for purposes other than they evolved for. Any other questions? Yeah.
I'm wondering whether there is contradiction in the priorities, the 10 priorities listed at the end,
Yeah.
between species specificity, killing 99 or over,
Yeah.
and self-sustainability of the parasite.
Yeah.
How-
You would rearrange those 10? Is that what you're telling me?
Not necessarily, but even contradiction in being able to self-sustain itself in the system and killing everything.
No.
It's specific.
No. No.
Yeah.
Did I say once eradication?
No, over
Kill everything.
99%.
Yeah, exactly.
Yeah.
Exactly. Because if you eradicate your host, you're out to lunch. Okay? And elm trees in North America, you can still find, I know very little about trees here, okay? Help me out. You can still find elm trees, but they don't get very old. Am I correct? You know, before the fungus knocks them back. So, yeah, you do have to have. The word eradication, I think I misled you because I gave the American chestnut tree, and I think that has been eradicated. You know, it's very difficult
There's
to...
still some in New York City. I've seen them growing.
Very small ones?
Yeah, the
And
small
elm.
ones.
Yes, and elm.
Big old ones.
Yeah, there are, but I mean, in terms of reduction, it's 99 point, right? I think so, especially American chestnut, I'm pretty sure.
Well, they die back and they don't.
Excuse me?
They die back and then they don't get very big. But they'll sprout up again from the trunk.
Yeah, and that's why you can have dry. I'm not saying that this is going to cause eradication. No, that's pie in the sky, and that's why maybe. And what she's saying there is that even with American chestnut and elm, it hasn't been eradication, but it's certainly drastically reduced, and that's why I said zebra mussels and quagga mussels are considered an invasive species because they cause harm. That's the key thing. There's a lot of exotic species we know do not cause harm. So if this thing worked extremely well, their levels would be so low that they wouldn't cause ecological perturbations. You see? Yeah. Yes.
I've got two questions.
Yes.
First one, when you talk about the tree,
Yes.
do you know where is the fungus?
The original fungus from Asia.
When there is no other tree, and all the trees are dead, and
No.
you plant a new one, the parasite is still
No.
there. So that means it's still present, and either in the
Oh.
soil with fungus, and I think that's how it works,
Yeah.
or it has another host. In the lake with mussels, if you have a parasite that is sustained, that stays there, even
Yes.
though you actually kill the mussel, that means it will have another host.
No, but there could, like eastern oyster is still on the Atlantic coast, but and there's been anthropomorphic pollution, of course, reducing its populations, and dermo reducing its populations, but it's still there. You see, so I'm not saying they're going to be eradicated. I'm not. They will still be there, and that will sustain the parasite. This is all the model.
That leads to my second question.
Yes.
If you kill 99% of the mussels and 1% survive, these ones are resistant.
No, not necessarily. No,
And
why?
if they are resistant,
Oh,
you
if there
actually
is-
need
Yeah.
a second parasite and do some sort of bacteria
Yeah.
to be able to.
But and I want you folks, look, you're marine people. You know more about the eastern oyster than I do. But I believe this is what, this is a hypothesis. This is my hope. Somebody tell me, has the eastern oyster, I know there's efforts, lab efforts, so to speak, aquaculture efforts to develop resistant strains to MSX. Have natural populations in 60 to 70 years develop resistance? Because you were predicting that would happen in 1 year. Can someone tell me about the eastern oyster? Because I don't think. Go ahead. Go ahead.
On the MSX side, actually, resistance came in a couple of generations. I mean, selection happened pretty quickly, artificially, by human-made, of course. Natural population today in Delaware Bay are about twice as resistant as the native Delaware Bay before the 50s.
Okay, so the resistance has developed.
Yes.
But there's still
The thing is, the
Yeah.
problem is that you have more than one single parasite selecting, of course, and you have some years with low pressure of selection, right? So oyster that are not necessarily very resistant come back to the system.
Yeah, you cannot. I want to really address your question. Thank you very much for really sketching that out. Resistance is always a factor. However, there's also a live parasite which is also adapting to its host. You see? Resistance, I'm acknowledging this is a very good point, okay? But the parasite is also evolving to keep up with it. No? Yes?
I'm thinking now about the mutation rate of the parasite. If the mutation rate is high, what are the chances that that parasite can find another host just...
Exactly, and that scares the hell out of me. And that's why, like, I'm getting a little out of my area because I haven't worked on viruses, but viruses have a much higher mutation rate than, let's say, some protozoan or haplosporidian like MSX. So I don't want to go there. You're absolutely right. In terms of choosing a parasite, I want the one that will have the least chance of having, but see, you can never say no. You can never, ever say no. But we don't, with the fungus that kills the elm tree, the fungus that kills different species, that kills the American chestnut, I don't see in the literature it spreading to other non-targets, you know, but you can never rule that out. You know what it's like? See, zebra mussels are marching across North America and causing all sorts of ecological perturbations. Industry effect, millions of dollars, chlorine being poured into pipes and power plants. That's an ecological problem, what's happening. So this parasite idea is a risk. You see, I'm weighing that against the destruction that is already occurring because they're proliferating across the country. So.
Is there a way to eradicate the parasite? You know, like if you create the you put the parasite in the lake, and then you have plan B to eradicate it as fast as
I don't think so. Let's be realistic. I don't think so. I think this is an introduction, and you better be damn confident. What I usually say is, because it's the number one thing. More money and more time will be spent on host specificity than anything else. You know, that's my, because I don't want another biocontrol, you know, disaster.
You say you don't want to go there with viruses.
Yes.
But do you take into account, or do you have any control that the parasite that you will introduce will itself carry viruses or associated microorganisms?
Yes. That is always a risk. In fact, some of my publications show the virus, the baculovirus, in the bacterium, and the bacterium is in the ciliate, and the ciliate is in the zebra mussel. So, no, that's a very good, I'm well aware of that, you know. Yeah.
So, are there certain, you need a parasite that has the capacity to be hypervirulent. Are there certain types of parasites that are more prone to this than others?
Well, you know, the examples I gave you were fungi, and I have not actually found, I think, any fungi in dreissenids. You know, I don't think anyone has found it. So there are many protistans, protozoans. There are parasites which use zebra mussels as the first host, and then they go on to infect the fish. You know, we're not going there. Yeah, you're smiling. Okay, exactly. We're not going there with those trematodes or whatever, you know. So, no, I don't. But again, it's not out of great depth of knowledge, but I just see stories about viruses and mutations. I wouldn't want to go there. Yes, you've had your hand up for a long time.
There's a few of my questions unanswered. Is there anything with synergistic effects that there may be a parasite that only becomes hypervirulent when the mussel is stressed, like when the temperature goes in a certain range or when some other chemical is present in the environment?
Well, you know, disease is affected by host and environmental conditions. So there is a range probably where the host, where the parasite could, if the host is stressed, for example, that's common, that disease is more prevalent. But I'm basing more of it on lack of coevolution, you know, that they're jumping. Now it's an intentional jump on my part, you know. So I don't know. Have I answered your question? I can't offhand come up.
Partially, but
yeah.
It's more that there's a lot of effects in a lot of these lakes. There's more than just the zebra mussels causing trouble. Particularly in parts of the Finger Lakes, there's a lot of other
invasives or?
There's not other invasives, but there's, well, if you want invasive plants, there's invasive plants that are providing a lot of substrate, but then there's other invasive things. There's the cycling of the lakes that they're kicking back and forth now between bloom and non-bloom in a couple of them.
Yeah, but you're asking, is there anything that could cause it to be hyperviolet? And it's too early, and I don't know. Nothing comes to mind. It's like
One
two.
more question.
Yeah. Okay. Yeah.
Is there something mechanically similar to parasporal crystals in relation to diatomaceous earth? Because I know they're both kind of serve as insecticides in some respect.
Say that again. I mean, these parasporal crystals are different kinds of proteins.
Okay.
And in fact, they're called protoxins. I didn't go into that. They're actually not toxic. And the reason they're so specific as BTI is there are certain organisms, when you put it in the stream, that can dissolve those crystals, and they become toxic. And that's why other organisms in the stream can eat those crystals, and there's no toxicity. So I don't know, diatomaceous earth.
I'm not totally familiar with the specific function of it, but it's mechanical. It's purely mechanical. It's not toxic.
Yeah, it's used, I think, to lacerate. Like if you have slugs and you put diatomaceous earth out, and they go through it, they can actually lacerate their bodies.
Right. I just want. I didn't know if there was a function similar.
No. Those parasporal crystals are attacking the midgut epithelial cells only after they've been dissolved. Black fly larvae, mosquito larvae have the enzymes to dissolve those parasporal crystals, and other groups do not.
Okay.
Yeah.
Let's have one last question.
So, thinking a little, let's go outside the shell here again. Have you considered, since you've found these specific species that produce parasporal crystals that work,
Yes.
have you considered taking the genes for that and splicing it into a bacterium that's already existing in the lake?
The parasporal crystal, well, the parasporal crystals were effective against black fly larvae, that was bacterium bacillus, and people have thought about that, taking those genes and putting them into diatoms or something, but there's been concern expressed here, and validly so, of doing things which are environmentally risky. And I never really want to put those crystals in algae and then throw it out into the lake. I don't want to do that because these organisms eat algae. So in a way, you say, hey, you have control all the time, but the whole GMO thing would take so long anyway. But it's beyond that. There's this inherent belief in me that it'd be even more dangerous than some of the things talked about today. Yeah, there's always. I just want to end this thing by there's risks, and you can't deny them. And sometimes you make a decision. And by the way, it's not me who's going to decide whether my parasite would be used. It's in regulatory agencies like the Department of Environment Conservation, the EPA. EPA has to decide whether this would get a registration label. Okay, so I present my safety data. Experts in safety look at it and hopefully say we're not taking too much of a risk. Thank you very much.
Okay, so for those of you who want to continue to talk to Dan, there's the Kubo room as usual for the seminars. So whoever wants to join us there, we'll see you there. Thanks for coming, everybody. Thank you for your attention.